All right, chapter 19. It's a little complicated, a little involved, and so we're going to break it up into several chunks. This chapter ought to put the cardiovascular system together for you. Remember that an organ system is made up of various organs. And so we looked at the heart, we looked at the blood that is found in the heart and in these blood vessels, and now we're going to talk about the blood vessels. First, we are going to talk about the various structures and functions of the various type of vessels. We will then move to the physiology of circulation. We'll look at blood pressure. In lab, we're going to take your blood pressure and talk about flow and resistance and how it influences that pressure. We'll talk about how that varies in different arteries, capillaries, and veins. And we will talk about the physiological mechanism by which blood pressure is regulated. Now, the flow part, okay, so that was blood pressure. The flow part also is controlled in terms of how much blood flow goes to a particular organ or tissue. And then we are going to get into capillary exchange. Now, this is the exchange of nutrients and gases, and it's cool. It's really cool. And this basic concept of capillary exchange will come back to us when we start studying the kidney. So we'll look at it in renal physiology. In lab, predominantly, you will be looking at and memorizing circulatory pathways. We do touch on them in lecture, so questions on them is still fair game, but you really are going to learn your blood vessels in lab. And that is how we continue with this, looking at the arteries and veins. So let's get started. So we are starting Unit 2 here. Again, going out of order, we are going to do Chapter 19 before we do Chapter 17. And Chapter 19 is the blood vessel chapter. It is really interesting. We will be applying some of that endocrinology that we looked at in terms of how blood pressure is regulated. We are going to start. So we are ready to get started on Unit 2. I hope that you feel okay about your exam for Unit 1. If not, let's meet and see what we can do to improve your scores. Again, A&PII 2 is very different from A&PII 1 in that we really are looking at a lot of physiology here. That being said, here in Chapter 19, when we take a look at the blood vessels, we will look at the anatomy first. As usual, that's what we normally do with each chapter. But once we go through the anatomy, we are going to then really focus on circulatory physiology. We're going to be looking at the relationship between flow, pressure, and resistance. And when we do, we'll be able to apply those concepts later. We will talk about the control of perfusion, blood flow through the tissues. We are going to talk about blood pressure, how it is regulated. And then another thing that we'll be able to apply later, especially in the kidney, is this concept of capillary exchange. Now, we looked at one of the hormones in the very first chapter of this semester that was ADH, antidiuretic hormone. It influences what the kidney does in terms of retaining fluid or eliminating fluid from the body. And so we will apply that to control. I'm just going to touch on it in this chapter, but as you can see, we're going to hit it pretty hard in Chapter 26. Now, in lab, we are really going to be looking at these circulatory pathways, both systemic and pulmonary, and then we have a few specific circulatory pathways, the one to the brain, the one involving the mesenteries, the digestive organs, and we'll take a look at fetal circulation as well. So buckle up. We're about to get started on a really cool chapter. This is a really excellent schematic of the circulatory system in general. It doesn't address whether we are looking at pulmonary or systemic circuit, but what it does include is the fact that our lymphatics drain into our bloodstream. And that's something we are going to take a look at in Chapter 20. But here we can see, as we already know, that the vessels leaving the heart are arteries, the vessels returning to the heart are veins. And in between, we have very important capillaries, capillary beds, and this is where substances are exchanged, nutrient molecules, waste molecules, and gases. The second thing that we can see on this illustration is normal blood flow is from the heart entering first elastic arteries, we'll talk about what those are, then branches off of those are muscular arteries, and as I mentioned before, the branches from those are arterioles or little arteries. We then get the blood flowing through the capillaries. From there, it drains into venules, small veins, and then into small veins, and then into larger veins, and return to the heart. So let's take a look at some details about how blood vessels are made, the different layers of them, and how different blood vessels are built differently. In your note outline, you see a drawing of what we might term a generic blood vessel with three distinct layers. The innermost one is tunica intima or tunica interna. The one outside of that is tunica media, and the most external one is tunica externa. Now, how these things are built and what tissue is found in these different layers is going to be different whether we're talking about an artery, an arteriole, a capillary, a venule, or a vein. So let's get started taking a look at how each one of those is made. Let's look at the artery first. Now, we do have all three layers in our arteries. They are also present in arterioles. But the difference between the artery and the vein is sometimes there are different layers. So in the artery, we see endothelium that lines the lumen of the blood vessel, and there is a subendothelial layer. In the artery, there is an internal elastic membrane, and that divides the tunica intima from the tunica media. The tunica media is made up primarily of a mixture of smooth muscle and elastic fibers. Depending on the type of artery or arteriole, it may have more smooth muscle and or more elastic fibers. And then again, different from the vein, the arteries have an external elastic membrane that separates the tunica media from the tunica externa. The tunica externa is connective tissue, a tremendous amount of collagen fibers, which is what gives blood vessels their integrity. Now, this is also where the blood vessel is perfused and innervated in the tunica externa, and we call that the vasa vasorum. When we compare the artery to the vein, we see that the vein does not have these elastic membranes. Veins are not very elastic at all, but they still do have the three layers: tunica intima, tunica media, and tunica external. One big difference is the tunica media in the artery is rather thick, where the tunica media in the vein is rather thin. Another difference between them we can see over here in the bottom of the vein drawing is veins have one-way valves, and we'll talk about the significance of those when we get into class. Up here in this illustration, you can also see a photomicrograph of a cross-section of these two blood vessels. The artery has lots of smooth muscle and tunica media. The vein has very little. The artery has enough smooth muscle that it's muscle-toned. It has a distinct shape, where the vein is rather flaccid. Capillaries are unique from these two because they only have tunica intima. They only have one layer of endothelial cells, simple squamous endothelium, and that is surrounded by a thin connective tissue layer called the basement membrane. Capillaries are so thin that they allow for rapid diffusion and exchange into the blood and out of the blood. Let's take a look at a blow-up of that first circulatory picture we took. Again, as blood leaves the heart, it does so through elastic arteries. These have the ability to maintain high pressure. They exhibit elastance, which means that they will return to their pre-stretched state. So a big bolus of blood can be pumped out of the left ventricle into the aorta. The aorta will stretch to accommodate that sudden increase in volume, and then the elastic fibers in that tunica media, and tunica media here is mostly elastic fibers, that will recoil and cause the aorta to return to its pre-stretched state. Branches off of the elastic arteries are termed muscular arteries. And again, they are named for the predominant tissue in tunica media. The further you get away from the heart here in the arteries, the more muscle, smooth muscle cells we see, and the fewer elastic fibers we see. And so these branches, we refer to them as distributing arteries. Branching off of these muscular arteries, we then see a lot of branches to very small vessels that are called arterioles. Arterioles have a tunica media that is all smooth muscle, and for that reason, this is where we see the majority of the influence of creating a larger lumen or a smaller lumen of these blood vessels. Vasoconstriction makes the blood vessel smaller, vasodilation makes it bigger. Now, that influences something we call resistance, which we'll talk about in Part 2. I'm going to jump ahead in your notes and just briefly talk about veins. The vein that drains the capillary is called a venule, postcapillary venule, and that in turn drains into, all of these will drain into small veins, which then drain into larger veins, which have a lot of capacity, Now, these high capacitance vessels means that this side of the circulatory system is what we call our volume reservoir. And that's in contrast to the arteries, which are pressure reservoirs. Think of the word reserve. Okay? So the arteries can store energy in the form of pressure. The veins store volume of blood. And in fact, the arteries at rest, you will only find 40% of blood volume in the arteries, where on the venous side, you'll find 60% of the volume. All right, let's move to another slide then and talk more about capillaries. Let's talk about a couple of characteristics that are true for all types of capillaries. And that is the intracellular cleft and the parasite. The intercellular cleft is a gap between endothelial cells. That means that membrane of the capillary is leaky. Substances can move out and into the bloodstream. Parasites are interesting. They are not as closely studied as a lot of other cells, but one thing is true. There's a little actin in parasites, so there are contractile fibers. And parasites can contract and help influence the size of a capillary. The most abundant type of capillary is the continuous capillary. They do have these intercellular clefts. There is pinocytosis across the endothelial cells. The only exception here for the continuous capillary would be the blood-brain barrier. Blood-brain barrier. You probably looked at that in, I hope, in A&PII in the chapter on the brain that shows that there is very little ability for substances to cross the blood-brain barrier from the cerebral capillaries to the cerebral spinal fluid. And this is what helps prevent getting noxious substances from the blood into the brain. So this is the area here that shows a tight junction and a tight blood-brain barrier. Next, let's talk about the fenestrated capillary. Now, these are really cool capillaries because they're even leakier than the continuous capillaries. They still have our intercellular cleft, but in addition to that, they have these pores or fenestrations that are holes, if you will, or windows that go all the way through the endothelial cells. Now, I use the word window because I learned as a little girl the German word Fenster means window. And that has always helped me remember that there are these large pores through which substances can cross. The final type of capillary we'll look at is the sinusoid capillary. These are found in organs where a lot of things have to move in and out of the bloodstream across the capillary wall. These are the leakiest. Not only do they have really large intercellular clefts, they also have enormous fenestrations. They're fenestrations, very large. And so you can see here that many things can cross from inside to outside of the capillary. When you add to that the fact that the basement membrane here is incomplete, there are actually gaps between the basement membrane, that makes it even leakier. The sinusoid capillaries are the leakiest capillaries in the body. Now that we've looked at the three different types of capillaries, let's look at a generic capillary bed. Now in this illustration, we can see that the arteriole ends up branching, and that blood will now enter the capillary bed. This is often referred to as the meta-arteriole, and beyond that is the thoroughfare channel. Why would blood only go through the meta-arteriole and thoroughfare channel? Well, that would be caused by what we call precapillary sphincters that would close off the branches from the meta-arteriole. If the sphincters are closed, if these smooth muscle cells here are contracted, no blood is going to enter that branch, that capillary branch. Here in the middle, though, you can see that these are open precapillary sphincters. The smooth muscle cells are relaxed, and this allows for this part of the capillary bed to be perfused. If everything's closed off, then the blood is shunted, we say, through the capillary and out to the venule. This is just a general look at flow through capillaries that are based on whether or not the arterioles are constricted or dilated. So at the top, our arteriole is dilated, and there is plenty of perfusion into that capillary bed. On the bottom, the arteriole is constricted, and this meta-arteriole will also be constricted, and that prevents perfusion into our capillary bed. This slide simply reinforces what I was talking about, how in systemic veins and venules, 60% of blood volume is at rest, is located there, where the arteries and arterioles and the rest of the blood vessels, this is where we find the remaining 40%. So again, our veins are volume reservoirs.